I think this is there in the clock, maybe. Okay, let's get rolling. Okay. So I'm going to take a quick reminder in case I forget at the end of class. So this is your first reminder that our basic procedure for this class is going to involve a short quiz every weekend that you'll complete by the time I see you on Monday. So that means you'll have a short quiz of 3 questions based on what we talk about in class today to do over the weekend. You get 2 attempts. I take the highest attempt. So I recommend you take the first attempt based on just what you actually remember with your brief, and then take the second attempt with your notes if you don't get the score you want. Okay. So you should be able to see that on Brightspace under Unit 1. And with that, we're going to get rolling with some content. Okay. So we're going to start by thinking about the human body. The human body is mostly made of water. We're going to be thinking about where we find different fluid compartments. So where we find basically water in different bags, because this is going to matter to us in the future as we start to think about different substances moving from one region of the body to another. Right? So we first want to define what our different areas are going to be. And then we're going to move into discussing the concept of homeostasis. So we're going to think about this idea of the fact that there are some things that we keep static in the body, some things that we control automatically that are important for your survival. So, as an anatomist, this picture hurts my soul, but this is a physiologist's simplified body plan, right? And the way it's drawn here is in order to highlight just the important bits for the purposes of thinking about where we find different compartments, different types of fluid that are the same and that can be different. So we're simplifying this to show relationships, to show where you need to cross a membrane, where you need to cross into a different cell type, and where you'd expect there to be a different kind of type of environment that you're working with if you're a little cell. Yeah. So it's a very round shape from an anatomy perspective. We've got just a square, but we've got all the tubes and things that we need here to understand this idea of fluid compartments. Okay. We don't need special regions for arms and legs and stuff because they're just going to be part of our general part of the body, right? So we're going to see no different cell types on here. We're going to be really hand wavy about things that are mostly the same. And we're just focusing on big relationships, big kind of types of environments here. Okay, so the first one to key in on that's going to be important is telling the difference between the external environment and the internal environment. So when we're thinking about this, we think about breathing, right? When in A&P 2, we start to talk about the lungs, we're going to be thinking about, okay, our external environment, the air, versus our internal environment. So thinking about the tissue of the lungs or the blood vessels in the lungs, right? We're going to have gases crossing between these two different types of environments, external versus internal. Okay. So when we have the external and internal environment, we're going to have a separation between them, right? So this is going to be an important separation that things like gases would need to cross. So this is our epithelial membrane. So epithelium, basically a cell type. Easiest one to think of is like kind of like your skin, right? Lining the outside of the body, right? Separating external from internal, but also lining some other places, right? So this is color coded in this image to depict external versus internal from a physiological perspective. So we can see that that epithelial membrane, that lining, isn't just on where you would think of as being the skin. We can also see it lining down into our lungs. We can also see it lining down here into our gut tube. So there are going to be some places that count as the external environment from a physiological perspective that you might not think of as external if you're just like lying in bed thinking about bodies. So we're thinking about a division between what touches the outside world versus what is fully contained and kind of controlled by your body. Okay. So we're going to have our external environment. I'm basically just going to redraw this picture and just focus on external, internal. Right, so external, outside. Okay. And then we're going to have a separation from the internal environment. So we have our epithelial membrane, right? So all the air outside of you, external environment. All the cells kind of inside your body are in the internal environment. But because your mouth is a hole, the air in your lungs still counts as part of the external environment because we're not controlling that, right? We can't control the composition of the air in the room. The air in the room touches the air in our lungs, right? So we don't have this kind of barrier protecting us from what might be in that air, right? The same thing is going to be true when we think about what's in like your stomach. Right? You've got actually a continuous tube when you think about the gut tube, right? From your mouth to your butt. Our gut tube is open at both ends, right? So it's in communication with the external environment. So that is also going to count as external from a physiological perspective, right? So that's why there's that big tube running through. So food in your stomach would count as part of the external environment. You don't suddenly become like 10% taco as part of your internal environment if you eat a bunch of tacos, right? It's not part of your internal environment until it crosses this epithelial membrane, which is going to be subject to some regulatory processes. This is also going to be true for your bladder. We've got an opening to the outside. If we have an opening to the outside, counts as external. So our internal environment is going to be the rest of our body. So this is going to be our immediate environment for most of your cells, right? So if you're just a generic cell, right, any random little cell, right, you are in the internal environment. So I probably ought to draw a big square body out around, right? So external was way out here. Then our internal environment is protected by this epithelial membrane. And then we have our cells somewhere in that internal environment, unless we happen to be like an epithelial cell making up the membrane itself, separating external from internal. Okay. So all of these cells are going to contain fluid inside them, and they're going to be surrounded by fluid. So we're going to have fluid inside the cell, we're going to have fluid outside the cell, and we're going to be interested in both. Okay. Now, depending on where our cell of interest is, the surrounding material is going to have two different names. So if you're just like a general cell somewhere in your arm, you're going to be surrounded by what we refer to as interstitial fluid. So that means inter means between. Stitial, I think of as like looking for that word tissue inside. So in between the tissues, going to be the fluid around most of your cells. But we have a very special type of fluid that's in your circulatory system, so basically in your blood. Right? So we're going to be more specific for in the cardiovascular system. It's our little loop of blood. Okay. So we have our blood cells bathed in a slightly different type of tissue of fluid called the plasma. So specifically surrounding those blood cells. So we've got plasma here. And surrounding just our generic cells would be our interstitial fluid. So this is how we're breaking down different regions of the body from a physiology perspective, thinking about different fluids, different compartments, different places we're going to have molecules, ions, gases crossing. So we're thinking about these different types of fluid, these different regions, because we're going to see, as a natural part of all the things that your body does, we have materials crossing in and out kind of all the time, right? So we're going to be able to absorb material. I'll talk about this a lot, especially when we, again, in A&P2, talk about kidneys. So this is a kind of for later thing. But absorption is going to be moving into the body, or we can put reabsorption in this category as well. right? So coming from the external environment into the internal environment, we have absorption or reabsorption, depending on exactly what organ system we're talking about. We also have the opposite direction. So you can see this arrow for secretion. So for secretion, that's basically out, right? So absorption in, secretion out. Our kind of general terminology here. And also, as we get into future organ systems down the road, don't have to worry about it quite right now, we're going to see another type of movement called filtration. So you can see that happening here as well. We're not going to get into the details of any of those processes for a while, but this is why we care about types of fluid. This is why we care about different fluid compartments when we're thinking about physiology, when we're thinking about bodies, right? Because we're going to need to pull nutrients, oxygen into the body from the external environment. We're going to have to cross from external into internal, and we're going to have to eliminate our wastes. So we're going to have to secrete or filter out certain things in order to get them out of our bodies. So when we think about those exchange processes, they're happening anywhere you have the ability to cross from one environment into the other. So in particular, we're going to be interested in this exchange between blood and the external environment. So that's this purple bluish stuff when we think about lungs, when we think about the GI tract, when we think about the kidneys. And we're going to be associating specific words with these specific processes in these different organ systems when we get there. So in the GI tract, in the gut, we're going to be thinking about absorption and secretion. When we eventually think about the kidneys, we're going to be thinking about filtration, secretion, and reabsorption. So if you're able, I'm going to ask you just a question to make sure everybody's with me. They can scan in the QR code or just answer on a piece of paper for yourself. But just a brief question about internal versus external. So we want to think which of these is part of the body's internal environment. What? Okay, great. Okay. So 3 of these answers were specific places inside the body that we made sure to point out were continuous with the external environment, so don't count as internal, right? So air in the lungs, food in the GI tract, urine in the kidney, all continuous with the outside, so still external. But extracellular fluid, even though it has the word extra in it, that ext kind of prefix really just means outside cells, not outside the body. So it's still the internal environment. It's the fluid compartment, the fluid that our cells are suspended in. Okay. So we're gonna think about how we name these different compartments now and think about what proportion of your body is really made up of these different types of fluid. So when we think about dividing these, we think about this thing called total body water, TBW, in your textbook. Literally just all the water in your body. No tricks here. All right. So our total body water is the total amount of water we have in our internal environment. And that total body water is what we're dividing down as we think about our different fluid compartments And these fluid compartments inside the body are also going to be separated by epithelial membranes. So an epithelial membrane, the lining, if you've thought about cell junctions before, they usually have tight junctions, right? So they're forming a seal. We're thinking like Ziploc bags kind of situation for our epithelial membranes. However, they do let certain things cross, right? So based on what proteins and stuff we have embedded in the cells of that epithelium, they're going to be able to selectively allow things to cross. They're going to be semi-permeable. So some things can cross, other things can't. So by the time we get to the end of this unit, we'll also be talking about different types of transport. We're going to be thinking about, does it require energy to cross this membrane? Does it not require energy? Does it require a protein to get across the membrane? Does it not require a protein? That's also not today, but that's where this is going. This is part of why we're talking about this right now. So our total body water, all the water in your body, a lot of dissolved materials in it. So we're also going to be thinking about gases dissolved in it, some ions dissolved in it, things like that. When we think about the total body water, it's all of it. So whether it's inside a cell, whether it's surrounding a cell, doesn't matter, it's part of your total body water. Next, we're going to subdivide that. Okay, so we're going to be subdividing into different compartments. When we think about the fluid versus the compartment, they're going to be named exactly the same, so we don't really have to differentiate that. So the first thing that we're going to divide our total body water into is our intracellular fluid versus our extracellular fluid. Great. We just saw, like most people were ready for this question, that the extracellular fluid is part of our internal environment, really is the internal environment that surrounds our different types of cells, right? So this is going to be abbreviated ECF, right? So when we're drawing cells later, I draw a little circle and I write ECF outside. I'm telling you that's the extracellular fluid. There's water, ions, stuff outside that the cell is suspended in. We also have fluid inside the cell that also has a name and an abbreviation. So that's our intracellular fluid. So inside the cell, we get an I. That's our ICF, intracellular fluid. Okay? So the contents of that fluid inside the cell is going to be a little different than the contents of that fluid outside the cell. We're not quite done with our subdivisions yet, though. We can leave the intracellular fluid alone. So we can leave the ICF, that intracellular fluid or that intracellular compartment alone, because we're really just thinking about cells containing cytoplasm, right? So basically that cytoplasm inside the cell is always going to be intracellular fluid, it's just what it is. But when we think about the extracellular fluid, we're going to be a little more specific because we've got two kinds of things depending on what type of cell we're interested in. Okay. I'm going to draw a little flow chart at the bottom here. Okay, so we've so far got our total body water. And if we're thinking about its subdivisions, we've got our intracellular fluid inside the cells. We've got our extracellular fluid, the other part. So that's anything that's outside a cell, so not contained inside a cell membrane. Okay? So part of our internal environment, but we've got two types and they can be significantly different, which is why they have different names. So we've got plasma. So plasma is a type of extracellular fluid that you find surrounding blood cells. And we've got our interstitial fluid. Be careful, it's an S in the middle, but otherwise it looks a little too much like the ICF. So the interstitial fluid with an S here, the between the tissues fluid, surrounds the rest of our cell. We don't differ between your leg cell, your arm cell, but we do differ between blood cell versus general body cell. Questions so far? case you're wondering about exactly how much water you would expect in these regions, here we have a breakdown. So we've got a textbook person. Textbook people are always imaginary. They're usually a 70-kilogram male, but we'll try to provide some variation on those numbers as we can and when it's applicable. But this works for our example here, right? So about 60% of body weight, about 42 liters, is how much water we would have in this hypothetical person. Okay? So of those 42 liters, we're going to split it between fluid inside cells, so 28 liters inside cells, and extracellular fluid, 14 liters outside cells. We can see here that that's 40% of body weight for the intracellular fluid, 20% of body weight for the extracellular fluid. And if you add those two numbers up, whether you do it by percent or whether you do it by liters, they add up to the 42, that total body water. So we got the two types, intracellular versus extracellular. And then we see the distribution of the plasma and the interstitial fluid. So those two numbers have to come up to our total extracellular fluid number because they're the two types of extracellular fluid. So we've got about 3 liters of plasma here and about 11 liters of interstitial fluid. So more interstitial fluid than plasma, but they're both important. They're different, even though they're both extracellular fluid. So periodically in your slides, you're going to see these checkpoint questions. So these are the types of questions that you might be using as you study, asking yourself, using those little writing prompts to make sure you understand what we did in that section of lecture and then in the textbook too. So I've highlighted one of them for you to think about and make sure you understand after class. We're not going to go through them in detail right now, but this is what you should be able to do. You can skip number 1 from this one. Well, we'll come back to absorption and reabsorption in detail later on this course. Now we're going to talk about homeostasis. Okay. So through the process of homeostasis, your body keeps itself at roughly the same temperature every day, keeps the pH of your blood at roughly the same number, right? Not too acidic, not too basic. Keeps the same amount of blood in your body, not too much, not too little. And it's really important that it does this because otherwise our organ systems and our cells don't have a predictable environment. And the way they function really depends on being able to expect the environment that they're in, right? They operate the way that they do when we talk about different cell types under specific conditions. Right? And if we really mess with one of these variables that's homeostatically regulated, we can't count on our cells working how they should. So homeostasis is going to be really important for our body functioning as well. So formally, right, official words, homeostasis is the ability of our body to maintain a constant internal environment. And so thought of that internal environment is all the fluid, all the stuff around our cells. So we're keeping that internal environment consistent in terms of its composition, but also in terms of things like pH, things like temperature. We'll go through a number of different, what we'll refer to as regulated variables in this course. So temperature is a big one and a kind of obvious one that you can sense when you're sick, right? You can tell when that one's kind of going wrong because you see a change in temperature and you don't feel well, right? That's already your body kind of struggling with that change. Volume, we're trying to keep consistent. If you have like really high blood pressure, or low blood pressure, that can be linked to the actual volume of blood in your body. You lose volume of blood if you get a cut and start hemorrhaging, bleeding a bunch, right? And we know from our practical everyday lives that that's a problem. Right? When we say composition here, we mean the composition of that fluid, the solute surrounding cells. So we're thinking, especially in this course, we're going to be thinking about like ion concentrations are going to be really important to maintain within specific ranges in order to keep things like nerves functioning correctly. To keep these variables consistent, right? So to keep these internal environmental characteristics the same from day to day, you're going to need a lot of different organs working together, right? You're going to have command centers in your brain working alongside receptors on your skin or in your different organs, sensing changes. And then you're going to have other organ systems involved in adjusting when we sense a change, when we sense a problem. Right? So we've got a real kind of whole body system here for the purpose of maintaining homeostasis, for keeping these variables constant. So when we disrupt homeostasis, we have a regulated variable that's important to us, important to your body, that we're trying to be consistent. If it goes far off enough, you might die. Certainly cause disease, depending on which of the variables it is and how far off from your set point or your desired kind of point we get with that variable. We'll look at some examples. The way that we're going to maintain homeostasis, if we think about a general type of process, is through something called a negative feedback loop. And in this case, I don't want you to think of the word negative as meaning down. I want you to think of the word negative as meaning opposite, right? So when we talk about negative feedback loops and look at examples of those, what we're going to be thinking about is when we see a change, a negative feedback loop pulls you in the opposite direction, so it pulls you back to normal, right? So whether we see your temperature go up, we bring it back down through negative feedback. But if you start to get hypothermic, right, if you get way too cold, you want to bring it back up to normal, that's still negative feedback. We're just taking it in the opposite direction from the change back to normal, back to our baseline. Okay. So when we think about negative feedback, if we wanted to graph it, right, we're going to have an X and Y axis, right? So on that X-axis, we're going to be looking at time, right? Because homeostasis is about maintaining variables over time, right? We want this internal environment consistent an hour from now, a day from now, a year from now. And our regulated variables, so basically the thing we're interested in is going to be over here on our Y-axis. Okay. So when I say that negative feedback means a change in the opposite direction, right, we're going to be cruising along with our temperature of 98.6, right, regulated variable. We start to get a fever, our temperature starts to go up. Your body has sensors that notes that difference. So this is our error signal, right? So we're sensing this change. Okay. And then the negative feedback is the process that pulls us back down, right? So this region where it came back down, right, this period of time, this is when negative feedback was happening. Works the same in the opposite direction, right? So if we then start to get way too cold, right, we still have an error signal, right? This is still a problem, we still sense it. And it still counts as negative feedback because we're just going the opposite direction when we bring ourselves back up to normal. So this is still negative feedback. So we're going to see a lot of curves just like this when you're looking at different physiological processes. You saw that we had a list of things and it was not a complete list of the things we're trying to keep consistent when we're thinking about homeostasis. In your textbook, they use a kind of analogy, thinking about like a car going up a hill, right? So you think about driving, turning on cruise control, but then you suddenly start to drive up the hill to school here, right? Your car would be sensing that we're putting a certain amount of gas in, right? We're keeping our speed the same here, but as we start to go uphill, our gravity, right, is going to start kind of pulling you back down backwards. So if you didn't add in more gas, you start to slow down as you're fighting against further force, right? So, you know, cruise control on, you're not going to notice on your speedometer, right? But there would be a slight error signal as we start to feel a little bit of slowdown. But then you increase how much gas you're putting in and you get back up to speed, right? So that error signal, there's the slowdown here, our regulated variable in this real life example is the speed. And we got a new word here. Okay. So we're also going to be thinking about set points. So the set point is this point on the y-axis that you're trying to keep stuff at, right? So in this case, it's that cruise control that you set to 60. In the case of your body temperature, right, it's at 98.6, you're trying to keep your body temperature at. That's the value you're trying to keep your variable at, okay? So our regulated variable is like kind of the more conceptual term, like speed, temperature. Our set point is a value, it's going to be like a number. And on the x-axis, we have time, because we're trying to keep this the same over time. Any questions about this basic part? Quiet when there are no questions, but also hopefully that means it's clear. Okay. So here we have that defined out in terms in life. So our regulated variable is the thing that we're maintaining. Speed of car. Something we didn't mention in some of the previous examples is your blood glucose. Like, so how much sugar there is in your blood is another regulated variable for homeostasis. This is why diabetes is a problem, and the inability to regulate the glucose concentration. We're going to keep blood pH, plasma levels of sodium, all consistent. Those are all examples of regulated variables. And then that set point is going to be the number. That's our expected measurement. So your body temperature, Celsius is 37 degrees Celsius. Our set point for blood glucose, your sugar level, should be about 100 milligrams per deciliter. Your blood pH is supposed to be at 7.4. Okay? So those would all be examples of our set points for these regulated variables. We saw my little drawing of negative feedback before, but here we have it defined and worked on the slide. So when we have a regulated variable, got a set point, we're cruising along. When we see it change, there it does decrease. When we see it change, right, change away from the set value, our system is going to respond and put it back to our set value. Right? So if it goes down, and your body pulls it back up, that's negative feedback. If it goes up and your body pulls it back down, still negative feedback, right? Negative just meaning opposite here. And the error signal is that difference that we're noticing, right? So over time, when we have these regulatory mechanisms activated, so when we're doing the process of homeostasis, right, our goal is to make that error signal smaller, right? So we notice that there's an error signal. We notice that there's a difference from where we want to be. And through this negative feedback loop, we're making that error signal smaller and smaller and smaller until we're back to our set point. So we said there are a bunch of different body systems involved in this regulatory process, in maintaining homeostasis. So depending on exactly what regulated variable we're talking about, that might involve different organ systems, but the general kind of process is the same. All right, so we need something to sense that we have a change, right? So in this example, we're looking at regulation of blood pressure. And this is something that you have to do every day. You're gonna have to do when you move on to lunch or your next class today, right? So if you go from lying down or sitting down to standing up, just like gravity tries to pull a car down a hill, gravity tries to pull your blood into your feet, right? You hopefully don't notice, although if you've ever seen your vision go kind of like gray or black, or you can see stars when you stand up, you were noticing this. But usually you don't, because this is a regulated variable and your body is supposed to really, really quickly adjust your blood pressure so that that internal environment stays the same and so that you don't just have blood in your feet, but not your brain. So we start with a stimulus. So a stimulus is like the reason that something's happening, right? So it's the thing that actually happened. So that would be like the standing up and causing the blood pressure to fall. So that's the thing that happened that messed up our regulated variable, moved it away from the set point. And then we have our first body system involved. So we're going to have some kind of sensor. Okay? So in this case, we have blood pressure receptors that would sense that the blood pressure has dropped in a lot of the important regions of your body, right? So like your brain, like your heart. So we sense this change, we sense the error signal, and then we're going to send a signal from that sensor to somewhere else. In the case of our blood pressure, we're going to have an integrating center in the medulla oblongata of the brain. So we're sending a signal going from your receptors through nerves up to your brain, where you have a region that processes different information from different parts of the body. Right? So we sense that our blood pressure has changed. This is a problem. But your brain also knows, like positionally, that you're now standing, right? So your brain can kind of help solve why this is happening and what you might do in order to fix it. So that's our integrating center. Then we're going to have effectors. So we're going to talk a lot about effector organs when we talk about what nerves do down the line. So an effector is something that does stuff, right? So in this case, we're looking at our heart as an effector organ. So in order to fix the blood pressure, to have an effect on the blood pressure, we're going to see our heartbeat stronger and faster. Right? We're going to push more blood. That's how we're trying to fix this error signal, fix this deviation from our set point for our regulated variable. Right? So this is the one that has an effect on our regulated variable. All of these processes in between, we would think of as signals, right? And then our final negative feedback response, as you'll see, is that change in the opposite direction, right? So we think about our little line, right? Our initial state, that first stimulus, was standing up and your blood pressure fell. And here at number 4, our negative feedback response is that your blood pressure goes back up. Okay? So that's what's happening here at the end. So here we have delineated out those different kind of parts of a typical regulatory response, a typical homeostatic response. So we've got our receptors, our sensors that detect stimuli, so our sensors that are detecting those error signals, that are detecting those changes. So you have receptors for sensing blood pressure, blood volume. Our baroreceptors are the ones sensing the blood pressure specific. You've got thermoreceptors sensing temperature. You've got chemoreceptors sensing chemical composition. In terms of integrating sensors, we're usually thinking about the brain. So this is where we're taking in a lot of information and sending out an appropriate response. Okay? So both sending a message from the receptor on your body up to the brain, and then sending a message from the brain down to, say, your heart. right? It's going to involve a signal. So we've got signal pathways. Usually when we think about signals, we're basically thinking about nerves, right? It's not the only type of signal you can have. You might put like hormones in this category as well. But if you're trying to think of a way to remember what we're talking about, it's nerves here. So this is allowing these different components to communicate. So we've got a signal going from the receptor to the integrating center. So like a nerve going from your blood pressure receptor to your brain. And then you've got a signal, an output signal coming out of your brain, going down to your heart, right? So we've got a signal traveling there as well. So usually this is going to happen through nerves or neurons. Could also happen through other chemical messengers going through the circulatory system sometimes. And then our effector organs, things that do stuff. So your heart is a muscle, specifically it's made up of cardiac muscle. Your other muscles also have effects, so you can generate like heat through movement of your muscles. Right? We all know that we get hot when we exercise, but that's what shivering is about too, right? You're trying to bring your body temperature back up by using your striated muscle. That's what we categorize skeletal muscle as. We've got smooth muscle around blood vessels, around your GI tract. So this can work too. Another effector organ that will sometimes be are glands. So glands can release enzymes, release hormones. So sometimes those are going to be involved as well in responses to changes in your body. They can release a lot of stuff, really. Okay. So a negative feedback loop is just a description of like a type of pathway and direction of travel. Right, so we've got our sensors sensing a change. They send a signal to our integrating center or whatever is doing the processing and deciding on our instructions. That integrating center, if it's doing negative feedback, if it's responsible for negative feedback, is going to send out a signal to the effector organ to do something to bring that regulated variable back to the set point. When you look at figures in your book, I'm just going to point out right now, this one isn't from your textbook specifically, but negative feedback loops in physiology diagrams are often dotted lines. So if you see a dotted line with an arrow, that usually tells you that this is a flow chart, but there's something that turns this off, turns this change off. That's why I've got a negative on it here as well. I'll point that out as it comes up within our organ systems too. So negative, moving in opposite direction from the change. We're making that error signal smaller, we're making the difference from the set point smaller, we're reversing the change from the set point, however you want to think about it. These are really all kind of saying the same thing. We're trying to go back to normal when we're thinking about a negative feedback loop. And this is happening constantly in your body. We're always kind of fine-tuning adjustments. You're always doing stuff that puts you into a different external environment, right? And your body's going to have to adjust to maintain homeostasis. Body temperature is a good example for the reason of you can feel it, and pretty obvious that body temperature goes both up and down, and both of those are bad. Right? So body temperature is a regulated variable. It has a set point of Celsius, 37 degrees. We've got sensors that detect body temperature. Your brain also processes information sensing that body temperature. Right? So the sensors go to the brain, tell your brain what the temperature is, and the brain forms a response to pull your temperature back to normal. So when we think about in real terms what that negative feedback perceivably is in our lives, right? If your body temperature goes up, right, the effect that brings you back to normal is sweating, right? So you have glands that release sweat, and then the evaporation of water off your skin hopefully cools you down and brings you back to the set point. So that sweat response from your sweat glands, those are your effector organs, are those sweat glands, and this is a negative feedback loop. This fact that you sweat to bring your temperature back down. And I mentioned shivering as well. also a negative feedback loop, negative feedback response. Right? If you start to get really cold, your body tenses up, you shake, you shiver, you get goosebumps. Actually, it's kind of neat because your body's trying to, like fluff up like a little bird or a little bunny or something, trying to trap air, trap warmth next to your skin. We're not really furry enough for that to have an effect, but it's in there, and at least the muscle movement does something for you there. So we bring our body temperature back up, largely through producing more heat through the movement of muscles there. Now, because we can have deviation in both directions, right? You can have things that make regulated variables go up, you can have things that make regulated variables go down. This means that we often have like opposite effector organs that do opposite things, right? So we have sweat glands to bring your temperature down. We have this shivering response to bring your temperature back up. And we can see that over the course of a day, as you move from cold room to hot room, go on a run, I don't know, have a job working in a walk-in freezer, right? You're going to be going through different environments. Both of these are going to be active. So a lot of our set points tend to also kind of incorporate like a normal range. So we think of this as dynamic constancy when we go up and down, kind of circling that set point, staying pretty close to it, but not necessarily exactly there. So the set point is the number we're aiming for, but we may overshoot or undershoot. And then we've got these antagonistic effectors that can bring us back to that middle ground when necessary. So we're at about time, but here's another example of what this process looks like, right? So here we're looking at blood glucose and its regulatory mechanisms, negative feedback loops here, right? So you see when you eat food, you absorb sugar from your food, right? You eat a donut, that sugar goes into your blood. Then we have a response from your pancreas releasing insulin, pulls glucose into your cells, so the blood glucose goes back down. And we've got a negative feedback loop turning this off once we're back to normal. Same thing happens if you don't eat, except we use different hormones. You can take a look at that and we'll pick up here next time.